So today's video is about development of eye. So first of all, you should know neural plate. So as you know that this is ectoderm, mesoderm, endoderm, this is notochord. So here basically the portion of ectoderm lying over the notochord becomes thickened to form a structure, and that thickened structure forming over a notochord is known as neural plate. So therefore I have written that the thickened portion of ectoderm lying over the notochord is known as neural plate, and this neural plate gives rise to neural tube. So this is the neural tube. So now I will discuss you about the divisions of neural tube. So here you can see that there are three divisions. The first one is the prosencephalon, second one is the mesencephalon, and the third one is about a rhombencephalon. So therefore that I have written that divisions of neural tube: prosencephalon, mesencephalon, rhombencephalon. Now, so here that this neural plate derived from ectoderm, and this neural tube derived from neural plate. So therefore basically this prosencephalon, mesencephalon, rhombencephalon, they are derived from both ectoderm as well as from neural tube. So therefore all the divisions of neural tube, they are neuroectodermal in origin. So now I want to discuss you about the optic sulcus. So this is a prosencephalon. So now initially if you see that in the prosencephalon there is no outgrowth, but now as you can see here that in this prosencephalon basically neuroectodermal outgrowth occurs, and this neuroectodermal outgrowth of prosencephalon leads to formation of optic sulcus. So see I have written that, so a neuroectodermal outgrowth of prosencephalon is known as optic sulcus. And why neuroectodermal outgrowth? Because this prosencephalon is neuroectodermal in origin. No, not only prosencephalon, in fact mesencephalon and this rhombencephalon, all divisions of neural tube, they are neuroectodermal in origin. So therefore optic sulcus clear. So now you can see, please see in this diagram that if you see that that this is the optic sulcus initial. So here this is the initial optic sulcus. Now here that this is the proximal portion of optic sulcus which is towards the prosencephalon. Now here if you see, as compared to the previous one, that this proximal portion has become narrowed, but this distal portion remains dilated. As a result, that this proximal portion is now called optic stalk, and this distal portion now known as optic vesicle. So therefore that I have written that the proximal portion of optic sulcus becomes narrow, and this proximal portion is towards the prosencephalon, and this narrowing leads to a structure known as optic stalk. Now again if you see that the distal portion of optic sulcus remains dilated, and this distal portion away from the prosencephalon leads to a structure formation, and that structure is known as optic vesicle. So proximal portion known as optic stalk and distal portion known as optic vesicle. Now you have to understand what is lens placoid. So now here you should know that that with respect to this optic vesicle, basically surface ectoderm lie. Now here if you see that similar in the case of neural plate formation, so here you can see here that this is the portion of surface ectoderm, so here lying over the optic vesicle. You can see here that this portion of surface ectoderm lying over the optic vesicle becomes thickened to form a structure, and that thickened structure formed over the optic vesicle is known as lens placoid. So here I have written that the surface ectoderm lying over the optic vesicle thickened to form a structure, and that thickened structure is known as lens placoid. Now so here you can see here that initially that this lens placoid lies exactly at the layer of surface ectoderm, but now if you see this lens placoid, so here separates from the layer of surface ectoderm, and here and that this the formation of lens vesicle. So here I have written that when the lens placard sinks below the layer of surface ectoderm, that means it sinks, so here separates also from this layer of surface ectoderm. Now that lens placard known as lens vesicle. Now here what is optic cup? So here now initially if you see the optic vesicle, it is only so here a single layer. But now if you see that this optic vesicle, it has two layers, see, one layer, first, second, two layer, and this double layer is due to differential growth of the optic vesicle wall. Why that means occurs? Because of differential growth of the optic vesicle wall. So basically that this double layer optic vesicle is known as optic cup, and this optic cup formation occurs simultaneously with the formation of lens vesicle. So therefore I have written that simultaneously during the formation of lens vesicle, the shape of optic vesicle changes. And now the optic vesicle has two layers, and that two layer optic vesicles is known as optic cup, and the region is also differential growth of the optic vesicle wall. So now the next part that I want to discuss is about only the separation of lens vesicle. So here if you see that initially that this lens vesicle is in contact with the surface ectoderm. But now here you can see that this lens vesicle completely separated from the surface ectoderm. And this happens during the 23rd month of intrauterine life. So therefore I have written that the lens vesicle gets separated from the surface ectoderm. Now the lens vesicle, so here becomes lens. Now this lens vesicle, that is, will become lens in future, but now it is completely separated. Now you have to understand what is choroidal fissure. So now initially if you see that this optic cup is not enclosing over the lens. Now also if you see, but now here if you see that this optic cup, so here has started to enclose lens vesicle completely from this superior surface and from both the sides also, but it has incompletely enclosed this lens vesicle from the inferior surface. So therefore that I have written that the margins of optic cup grow over the superior and lateral basically sides of the lens vesicle completely, but except the inferior side. See that this inferior side is empty because complete enclosure doesn't occur in the inferior side. So here now if you see that as a result that with rest in the inferior side, with respect to this lens vesicle, that is an opening created, and that opening is known as choroidal fissure. So this is what I have written, that choroidal fissure. So this is choroidal fissure. So now I am going to discuss you about the primary and secondary fibers. So now this is a lens vesicle. So this is lens vesicle. You can see here that initially, so this is the anterior wall and this is the posterior wall and this is the cavity. So now initially if you can notice that both the anterior and this is the equatorial region, so both the anterior and posterior wall, they contains a single layer of cuboidal cell. So therefore that I have written that initially in the lens vesicle, both the anterior and posterior walls contain a single layer of cuboidal cells. So this is the initial condition. But now here you can see here that this is a posterior wall. So here basically the cells of the posterior wall, so here converted from columnar, from cuboidal to columnar. And also you can see here that initially the cavity of lens vesicle was large, but slowly due to that is growth or elongation of these columnar cells or the conversion of cuboidal to columnar cells, that this cavity of lens vesicle is started to get obliterated. So therefore that I have written that the anterior, but till now this anterior wall is same. The anterior wall remains cuboidal, but the posterior wall cells converted into columnar cells and they are slowly obliterating the cavity of lens vesicle. Now here if you see that this cavity of lens vesicle has been completely obliterated, and this posterior wall cells secrete fibers, and this posterior cells, so here secrete fibers, and now the primary fibers form. So here you can see that finally the cavity of lens vesicle obliterated, and the posterior columnar cells secrete fibers called primary fibers. Now about secondary fibers. So basically you can see here that this is the equatorial region, and here that, and this equatorial cell regions also secrete fibers, basically secondary fibers. So here I have written that the cells of the equatorial region also secrete secondary fibers. That's all about today's video. So today you have learned about what are the divisions of neural tube, important points, what is optic sulcus, what are optic vesicle and optic stalk, and what is lens placard, and what is lens vesicle, and here basically optic cup, choroidal fissure, primary and secondary fibers. That's all about.